Can you continue, sir? So as I already informed about the insect vectors, mainly that the psyllids, plant hoppers and leaf hoppers are major vectors. But apart among these, only one insect, that is Oraceus alpicinctus leaf hopper. It can able to transmit nearly 99% of phytoplasma to the sesamum plant. We have observed that one. So apart from that, recently we have noticed one more leaf hopper, that is Hicimonas, that is last one, Hicimonas phycetus, that also transmitting the phytoplasma in sesamum. So apart from that, other researchers also observed that Oraceus cellulosus also able to transmit the sesamum phyllodes. So recently we have encountered mirid bug. So wherever that sesamum phyllodes infected plants, we are observing more number of mirid bugs. That symptom also is giving. So we are also in the process to identify whether this mirid bug also able to transmit the phyllodes or not. So we are also determining the infection in the sesamum. These all three pictures are Oraceus alpicinctus, and the last one only Hicimonas phycetus. So it was confirmed by both allowing the viviparous means acquired Phytoplasma insects into the plant, healthy plants, and also through molecular also we have identified that presence of Phytoplasma both in insects and also in the plants. Next, please. So we want to know the different strains or subgroups within the Phytoplasma. So we have taken nearly 92 sequence from the public domain. So we have grouped in the neighbor joining method. So in that we have collected sequence from right from Egypt, Iran, India, Taiwan, Turkey, and Vietnam. So around 8 countries we have taken the sequence. We have made the neighbor joining method of phylogenetic tree. So here we found three subgroups. Subgroup 1 is 16SR1 and subgroup 2 is 16SR2 and 9. So the first group 16SR1 is found mainly from the sequence of India, Egypt, and Thailand. So it categorized here. First group. Of India, Egypt, and Thailand. Second group starts from this here to up to that end. That group's second subgroup of strains of 16SR2. So that from the in different countries like India, Iran, Taiwan, and Turkey, Oman. Likewise, the third subgroup of the strains, that is 16SR9 strains, only present in Iran and Turkey. So from this, we clearly able to observe that different strains of Phytoplasma and also the diversity we able to find. Only this subgroup 3 or the strain third 16SR9 is not present in India. Next, please. And we have tried, because mostly we are telling about the transmission of Phytoplasma by vectors. So culturing of vectors in the lab condition or artificial condition is a little tedious process, very difficult to culture the insects. So we want to know other means of transmission. We tried with the different methods to transmit the Phytoplasma to healthy plants by Dardar method and also grafting, and infected seeds. The seeds produced from the infected plants also we have taken. That also we have tried. Even we have tried with the sap inoculation also. So among these 5 methods, we have found only vector transmission is able to transmit 99% transmission, and followed by grafting. So nearly 80% can able to transmit the Phytoplasma by through grafting. But in data only 20% able to transmission of Phytoplasma observed. But nothing happened in sap inoculation and also infected seeds of sesamum phyllodes. Next, please. So here some work we did, and I collected from we have ACRP centers. All India Coordinated Research Projects on Oilseed Crops. We have around 40 centers. There we are used to send our material to screen for resistant any materials like genotypes or advanced breeding lines. Here we also given some activities to see the cultural methods and other methods which effective against the leafhopper and also Phytoplasma in sesame. So here we found that mainly we all know that crop rotation. So wherever we are doing crop rotation and then let's say sowing of sesame. So mostly it's sown in cultivated in Kharif season. Nowadays it's shifted to Rabi also, even summer also. If you go summer, you have to start in the month of January. If you start sowing in the month of January, the infestation or in the infection of Phytoplasma will be very less compared to other months. Later slide, I will explain about the details. So apart from that, we have tried with the different intercrops and we found only this PGNP with the sesame 1:1 ratio is found effective are able to prevent the entry of hopper that automatically prevent the infection of Phytoplasma. So apart from that, we are also other works we are doing that water crop with the different crops. So most of the viruses are we used to with the border crops. That also we are trying with the different border crops and also the intercrops to avoid the infection of Phytoplasma through vector. And we also tried with the different insecticide seed treatment along with the application of neem oil. We found imidacloprid seed treatment with the neem oil application effectively we can control the leafhoppers and also Phytoplasma apart from the application of Karanji oils and also the NSK applications. And we also tried some antibiotic means of control, spraying of tetracycline and oxytetracycline along with insecticide. Not alone, it's not effective controlling of Sesame floridi. So we tried with some insecticides, even seed treatment with insecticide and followed by insecticide application. It's effectively control the Phytoplasma. And similarly, we have tried with seed treatment with imidacloprid followed by imloc application or thiamethoxam. Either one is effective against the control of Sesame floridi. Next slide, please. So here only since IIO are having more germplasm of sesame, so we started screening and we want to know which season since it's not feasible in screening under artificial conditions since we required more culture of leafhoppers. So we want to know which season is infection of Phytoplasma is occur more. So we want to know that one we have shown the sesame in 3 different or 4 different sowing date July, August, November and January. We found the November and July one only had highest percent infection of Phytoplasma and also leafhopper population is more. So we have selected only the July zone one only since the population of hoppers and also the Phytoplasma incidents are more high. So we have taken for further studies and 2019. Next, please. So we have taken around 104 germplasm to screen against the Sesame floridi and also leafhopper responsible for the transmission of Sesame floridi. Here, out of 104 germplasm, only 3 germplasm, which is highlighted in blue color, had resistance against the Sesame floridi and also had very less population of leafhopper. That genotypes we have taken for further confirmatory trials for next 2 years, like 2020 and 2021. Next slide. So here we have taken only the 3 genotypes along with susceptible check and also resistance check. Here we found only these 2, GTG 30 and third one having less resistant, I mean less infection is below the 10%. That means it's a resistant category. So we have confirmed that also we have confirmed through molecular also by using 2' primer. Next slide. We have confirmed that the presence of the Phytoplasma in the susceptible genotypes only, not in the resistant genotypes. Next, please. So with this, many works are required to manage this Sesame floridi, especially since it's a old long issue for the control of this phytoplasma in sesame. So these are the main future works to be conducted, especially the survey where the sesame growing areas. We have to document the regions or seasons where the phytoplasma is free from sesame, and also the vector phytoplasma interaction and vector plant interaction also need to be studied properly. Apart from that, epidemiological studies also required to study, and also the alternate host we have to identify. We have identified only few alternate hosts, especially transmission of sesame phyllode. Phytoplasma is infecting more host, but especially that sesame phyllode, that group I said na strains 16SR 1, 2, and 9. That group is whether is there in other plants or not. Which one is serving as a reservoir for the transmission when the sesame was not there in the field. So that study also we have to take, and especially development of resistant varieties. Mostly they have screened in the open field conditions. So we have to confine in the artificial condition to release the phytoplasma occurred insect and feed on the genotypes or germplasm. Then we can get the proper resistant source for the management of sesame phyllode, and also incorporation of transgenic resistance in agronomically or yield superior cultivars. So this also possibility, and finally that's sustainable management of phyllode. So we have one collaborative research project, sir, with other universities. We have ASRP systems. So we are involving our Virudhachalam also is one of the ASRP centers. We are involving them also for this activities, sir. And many universities we are involving, since this is a ICR given guidelines to have a collaborative research work with all the possible collaborators in India, like that, sir. We have started developing the projects to work on these issues, sir. That's all about this work at ICRI or Hyderabad. Thank you. Oh, very good. Very good, sir.
What is the right? We have to
Yeah.
aim to follow this.
Follow this, yes, sir. Yes, sir.
The salivary glands exclusively.
Very good.
If we understood that even within the salivary glands, the phytoplasma is present.
Yeah.
Also, is trying to do some further confocal studies.
Okay, sir.
To see how it is getting localized in the salivary glands.
Salivary glands. Okay.
And I think she has documented many other paths,
Very good.
including papaya.
Yes.
She did a lot of sequencing
Okay.
for these strains.
Okay.
Phyllode strains. I don't know whether which strain. There were still in 3 strains. We don't know. Strain not the first strain. Okay. So,
Okay.
yeah. So we took a lot of samples from most of the tracks of the injury.
Okay.
Yes, injury is grown around the year.
Around the year. Okay. Okay, sir.
This is eroded. It is grown in 3 or 4 seasons.
Okay.
And Phytoplasma is again grown in 2 seasons, I think.
Okay.
Then the Tanjavur and other region, rice fellow
Ginger is there.
So many seasons are there.
Okay, sir.
That's why we are trying to understand the population dynamics also.
Okay.
I think I can talk more. If you have any questions, we ask.
Sit and I can ask. Okay. We have taken some, we want to confirm through morphology and also molecular that species. We have separated So mainly these two species, what I said, Voroses albicinctus and also the Hicimonas spicetes. Nowadays that Hicimonas spicetes population is more than the Voroses albicinctus. So uh,
It's known as predominant, but
predominant.
transmission is also more severe.
More severe, yes.
More severe.
Yeah. So you can try with even merit bug, I told you. So I do not know whether it's merit bug problem is here or not.
Blue Phytoplasma merits.
Yes, sir. Phytoplasma merits. That I have sent for identification, sir. Possibly is there, sir. Yeah, similar. Yeah, same one. Yeah. Okay. Pagas. Okay.
Blue pagas, actually, it's a good predator.
Okay.
When predators are not available, it will survive by feeding on the plant.
Okay.
So that's why it is blue bite. It is called blue phytoplasma.
Okay.
But unfortunately, I clear from Indira Gandhi that it is causing severe damage.
Severe damage, sir. More than 80%, sir. More than, yeah.
Okay. Tomato ecosystem, mostly
Okay.
in most of the succulent
Crops, sir.
Nistioporis, the presence of Nistioporis can be seen.
Okay.
It's a good predator of Tuta absoluta.
Absoluta.
So, there's a lot of advantages. But anyhow, recently I started getting from injury workers causing lot of damage to.
More damage, sir.
Yeah. So then we have to see at what level it is transferring the load and other things.
Yeah, we have to study, sir. We have also initiated that work to see in the insects also. So separately, the transmission studies also in the lab conditions. Okay.
Possible, I will also try to collaborate with you, Dr. Boopathi.
Sir, sure, sir. Yeah.
I'll try.
So even we are planning for a effector biology also, sir. So okay. 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 Okay, sir. Okay. Okay, sir.
There's no information on the transcript of data, so this is specific.
Okay.
Data. So we were able to exercise the cellular gland.
Okay.
Most of the microinsects, is the microdissection and the transcriptomics.
Okay, sir.
So we did the kind of work. But that's why I insisted Sadhana also to go for salivary gland specific PCR.
PCR, yeah.
In that also we get the phosphorus regards and vasculature. We sent those data for sequencing of them.
Okay.
Probably, I think some line of work we can also plan to.
Yes, surely, sir.
So I discuss with you later and see any some places students we can collaborate and we can exchange.
Exchange also, yes, sir.
Can allow to come over there.
And, yeah, that will be good, sir. Yeah. Even many we are used to say only insect maximum transmitting the phytoplasma. Apart from that, hormonal changes also is main. So one more studies also is there. So because it's flowers only turning into the green color.
Yeah.
So possibly it's a hormonal changes that also reports are available. So in that also we have included this one objective to see the hormonal changes also, sir, if possible. That all. Hormone changing, yes. Yeah.
Or the feeding the saliva injected inside, phloem.
Phloems.
Some effectors.
Effectors also, yeah. That's what.
malfunction. So that, the plants is being hijacked by the leafhoppers
Leafhoppers.
for their favor.
Yes.
Maybe some interesting term or.
We also started on the effector also, sir.
Oh, okay, okay.
Some work, yeah.
Okay.
Last 1 year we have started working on the effector also.
Okay.
So some work is going on. Final, maybe within 1 year we may get that some result towards that one, sir.
Oh, yeah.
Mm. Mm. Pungam oil. Pungam oil.
So North Indian, the Hindi name is Karanch.
Karanch.
Okay, Karanch oil, which highly useful oil. Earlier it was thought that, Karanch oil is causing the carcinogenic.
Carcinogenic. Yeah. Mm.
But later it was proved that it is not causing any carcinogenic, not at all a carcinogen. And, now people widely use. So even extraction units are available in Dharmaburi. Okay, so the forest department is excluding Hosur, near Hosur, Sulagiri. There is a extraction unit for this Pungam oil. Okay, so the Pungam, oil, all trees cannot produce Pungam oil. Okay, only the regions where the night temperature is low, there only the oil accumulation will be low. Okay, even in front of the students' canteen, am I right? University canteen, there is some Pungam tree. Am I right? So a lot of insect samples, leaf blotches, then, the mites, all those things you see there are a lot of feeds are safe. But if you take those feeds and extract oil, you won't get oil. Okay, only in cooler regions. So that's why the factory is running in Dharmaburi, where because the Dharmaburi and other regions of the western Tamil Nadu is sitting on a higher environment. Am I right? So that's why the night temperatures are low and the temperature variations are high extreme temperatures. Is favorable for income oil production. Right. Any other questions? Please. Also, for the information on the house, Pungam oil is a good, what do you say, synergist.
Synergist effect.
Good synergist. Now, if you mix the insecticide with the Pungam oil as a tank mix, if you go, you will get good results. But if you put MF4 in the paper. Any other questions? Wind up. Thank you. So thank you, Dr. Pujari. It's a great time.
Thank you, sir.
Hope, you will induct one or two students from this campus.
Yeah, sure, sir.
And Dr.
Yeah.
We have good students, good strength of students.
Okay,
They
sir.
don't know what to do. If you give some topics, they will do the best. Okay.
Okay, sir. Okay, sir.
Thank you. Thank you, guys.
Thank you. Thank you, sir. Thank you to everyone.